Hello and welcome to In the Lab with Bioreach Protocol 1 Autoinduction Scale-Up. This video will serve as a helpful visual guide to the written protocol available for download on the Bioreach Cambridge website. Why do we need a protocol for autoinduction scale-up? Well, we need this protocol so that we can express the recombinant DNA we transform into our bacteria. To produce recombinant proteins, bacteria like E. coli must first be transformed with a plasmid containing the recombinant DNA. They are then grown to sufficient cell density before inducing the expression of the protein. In this protocol, we use an agar medium that not only contains all the nutrients required for bacterial replication, but also a chemical that will automatically induce the expression of your desired protein. Our plasmid includes a lactose operon, also called lac operon, that allows for autoinduction of recombinant enzyme production. The lac operon includes the lac promoter, the lac operator, the gene of the desired protein, as well as a terminator. The main source of energy for E. coli is glucose, but they can also metabolize lactose when glucose is not available. When lactose is metabolized, the repressor on the lac operator is removed, which allows the expression of the desired protein. By optimizing the ratio of glucose and lactose in the agar medium, we can combine the bacterial replication stage and the induced expression stage in one step. Sufficient glucose allows the bacteria to grow to the optimal cell density before the glucose is exhausted and lactose must be metabolized, inducing protein expression. The biomass can then be easily collected by scraping the agar plates and the desired protein can be extracted. For this protocol, our protein of interest, BST, is linked to the fluorescent protein M-cherry so that the protein expression can be easily confirmed with the naked eye. Here is everything you will need to successfully prepare the full XYT medium, express your protein of interest, and assess your collected biomass. You can pause the video here to make sure you have everything you need to begin. For the first day, we begin by preparing the 4xYT autoinduction medium. We first weigh the nutrients and salts required for bacterial growth and then transfer these to a heat-safe 1 liter glass bottle. We then dissolve the added chemicals with half a liter of distilled water. To avoid contamination, this medium should be autoclaved. We also need to prepare 3 additional solutions, each in 250 ml glass bottles. Shown here, we are dissolving 10 grams of glucose in 100 ml of distilled water to prepare a 10% glucose solution. Using the same methodology and in a separate bottle, you should also dissolve 10 grams of lactose in 200 ml of distilled water to prepare a 5% lactose solution. The third solution is prepared by adding and mixing 50 ml of glycerol with 50 ml of distilled water. All 3 solutions should be autoclaved to avoid contamination. For day 2, we begin by melting our agar medium. In this video, we use a hot plate, but you can also use a microwave. To heat the medium evenly and prevent it from boiling over or burning, it may be helpful to cut the agar with a sterilized metal stick or rod. Make sure the lid of the bottle is loose or removed entirely and keep an eye on the medium at all times. Once the medium is completely liquid, remove it from the heat and allow it to cool slightly so that it is safe to handle and no longer boiling. Now, to reconstitute the medium, add 12 ml of the sterile 50% glycerol solution, 40 ml of the sterile 5% lactose solution, and 5 ml of the sterile 10% glucose solution. Bring the reconstituted medium to a total volume of 1 liter by adding approximately 440 ml of sterile water. Using either a lit Bunsen burner or a sterile flow hood to prevent contamination, prepare your agar plates. Add the antibiotic marker to your agar medium and mix. In this case, we're using a 50 ml Falcon flask to ensure a consistent volume of medium in each plate. For each 9 cm wide plate, we will pour 20 ml of 4xYT autoinduction medium. Pour the medium carefully and try to avoid making any bubbles. Now, allow your plates to solidify in a sterile environment. This should take between 15 and 30 minutes. Once your plates have solidified, spread your transformed bacteria. Here, we are using an overnight culture of E. coli containing a plasmid for BST M. cherry. For each plate, evenly spread 200 microliters of bacteria with a sterile rod. This procedure should also be done around a lit Bunsen burner or in a sterile floor hood. The incubation time required for protein expression will vary based on the incubation temperature. At room temperature, 40 to 72 hours are recommended. At 28 degrees Celsius, 16 to 64 hours are recommended. At 37 degrees Celsius, the bacteria should be incubated overnight for 12 to 18 hours. Prior to collecting the biomass, you can confirm that autoinduction has been successful based on the color of the bacteria. If the bacteria is pink when scraped off the agar, then BST M. cherry has been expressed and can be collected. If the biomass is yellow rather than pink, you may need to incubate it for longer to ensure that it has sufficient time for autoinduction. You can collect the biomass in a microfuge tube or any other suitable container by scraping the top layer with a glass plate. It is important to keep consistent yet gentle pressure to avoid cutting into and collecting the agar beneath the biomass. Once you have collected your biomass, you can prepare a functional extract of your enzyme through the protocol found on our website. We will not be showing the crude extract preparation in this video. Instead, we will provide you with a method to quantify your enzyme production using a spectral photometer. Using the spectral photometer, prepare a blank by adding 1 ml of PBS buffer to a cuvette. Once you've read the blank, prepare a separate cuvette. Add a small amount of biomass and suspend it in 1 ml of PBS buffer. You may need to pipette up and down several times to ensure that the biomass has been evenly and completely suspended. Using the spectral photometer, read the sample at optical densities of 700 and 587 nanometers. The OD 700 value quantifies cell density and should be at around 0.6. The OD 587 value quantifies the amount of M. cherry. At an OD 700 value of 0.6, you should expect an OD 587 value of approximately 0.8 for full expression of BST M. cherry. This video has shown you all the steps involved in the autoinduction scale-up protocol. You should now be ready to start bioproducing your own recombinant enzymes in the labs. Please keep in mind these helpful tips. When melting the agar, it is important to continuously monitor it to prevent boiling over or burning. It may also be convenient to break up the solidified agar into several sections in smaller bottles so that you only melt the amount of agar that you really need. The amount of biomass increases proportionally with the surface area of the agar plates used. If you wish to produce enzymes in larger quantities, then it would be useful to maximize the surface area of your autoinduction plates. The spectrophotometer step is not essential to the production of your recombinant enzymes, but rather one way you may consider quantifying your enzyme production. If you are ever unsure about something, do not hesitate to ask for assistance from a more senior lab member or drop us an email. Have fun and stay safe in the labs. Thanks for joining us today and see you again soon.